He's assistant professor in chemical engineering at the Israel Institute of Technology in Haifa.
Thank you. Yeah, thank you so much. I'll get directly to the point. I think one of the things we always get are, there's one rule, right? Before you give a talk, you never look at your emails. And I did, and I got another reminder of the importance of the work we're doing. This is a patient who has been reading our work and emailing me over the past half year, about twice a month, showing me new papers in the field of nanotechnology. And today at 2:24, as I was checking my emails, he emailed me his last one after the previous one showed me a new study about nanotechnology in medicine. And this says, it's 2:24 today, "Bye, Avi, my time is near." So I think so many patients actually depend on us developing new technologies and really giving more promise to their conditions. And I think that's one of our obligations is to actually translate these systems. And I wanted to start actually with this image from Harrington's paper. This is a paper from 2001, which I think opened to many of us the field, showing that nanoparticles can actually target tumors in a rather efficient manner. 3 different types of tumor: a tumor at the base of the tongue, a patient with breast cancer and actually metastasis, and a patient with lung cancer, metastasis in the spine, and also in the bone marrow. And while different types of cancer, all 3 patients were receiving 100 nanometer particles with a contrast agent inside of them. The nanoparticles are injected intravenously. They circulate in the blood, and they actually target the tumor site. And at the time, we thought that all tumors have these leaky blood vessels, and nanoparticles can extravasate out into the tumor site and in an effective manner. And I think today we know, after more and more information is actually being generated, is that this may vary from patient to patient, but for those patients that have an increased permeability of the vasculature, using nanotechnology can grant a major advantage in their therapeutic outcome. And based on this phenomena, more than 80 nanomedicines, nanotechnologies have been approved already by the FDA and other regulatory bodies. And namely, we should mention the first one to reach the clinic is Doxil, invented by Professor Chezi Bernholz, who's sitting here in the audience. And that marked actually how nanotechnology first can do 2 things, achieve 2 major advantages. 1, reduce side effect, namely cardiotoxicity of the doxorubicin, and 2, improve the therapeutic outcome of the patient. And the evolution of these systems has been into loading, if here we have one medicine inside the nanoparticle, recently a liposomal system with 2 medicines inside a single nanoparticle, a single liposome has been approved, targeting 2 different pathways inside the cell. And some of the other nanotechnologies are based on proteins, Abraxane, and I think what really marked the change in our field is the approval early this year of Onpato, the siRNA lipid-based nanoparticle for treating liver disorders. So we're going to more and more sophisticated systems and mechanisms. The field really evolved a lot throughout these years, but still many patients actually enjoy even the first technology that entered the clinic, I think 1992, right? If I remember correctly. 1995, sorry. So I'll be talking about liposomes and lipid-based vesicles, and specifically I want to go back to that image that we saw first with 3 different patients. And if you swap the contrast agent with a medicine, you can, of course, improve the therapeutic efficacy. But we actually wanted to look at something else. We wanted to ask which medicine would be best for each cancer patient before we start treating. Can we use liposomes for diagnosing the personalized medicine for each and every patient before starting a therapeutic cycle? And the field is called personalized medicine, and we all know that it's really hard to determine ahead of time what a patient will or will not respond to. And even here, if we check this room, most likely just in pain medication, each one of us ran this clinical trial on him or herself, testing which pain medication is best for us, the fastest efficacy, the lowest side effects, and we continue with that. But when we talk about cancer, they're trying the trial and error is actually much more painful to the patients. And I always, when I think about personalized medicine, I think of House, right? I mean, Dr. House is personalized medicine at its best, looks the patient in the eye and says, it's lupus here, right? While everything else indicates, or the charts indicate something different. But unfortunately, he's not in the clinic and can't really help us when we need him. In fact, when we take the best studied cancer, breast cancer, with the best technologies out there, still 1/3 of breast cancer patients are receiving the wrong medication, a medicine that doesn't affect their own tumor. And that's devastating, of course, to the patients themselves, to the caregivers, their families, and everyone that's in that circle. And also the pharmaceutical companies, I believe, today want to choose in a better manner, want to give medicines that will actually help their patients. And when we talk about personalized cancer medicine, the dogma is usually to take a biopsy from the patient and then sequence it or look for different biomarkers, and based on that, run drug selection. And that really advanced medicine greatly, and we heard about different biomarkers and how they can actually improve the therapeutic efficacy. However, in breast cancer alone, we know that pretty much reaches a plateau of the 70%, where beyond that, not all patients will respond according to what their genetics actually predict. And the reason may be that we don't understand the genetics well enough, or that that other effects beyond genetics may be affecting how we respond to medication. So a second set of technology kicked in. Here a biopsy, again, will be taken from the patient, and the cells from the patient will be screened in a dish, and now inside the dish, these patient-derived cells can actually be treated with different medications, and we can look what the cells from the patient will or will not respond to. However, in the dish, they respond differently than they respond inside the body. So an advancement of this technology was to take the patient-derived cells, grow them in a mouse, and now treat the mouse with these with different medications and look what the patient-derived cells will actually respond or not respond to. And this actually is a very effective technology. However, there is one limitation. If this takes 4 to 6 weeks, this usually takes 4 to 6 months. So it's just too long for many of the patients when you come to screen the medication. This is where we thought nanotechnology may be able to actually compete with the existing state of the art. Specifically, what we asked is, can you use nanotechnology to screen medications inside the patient's own body before you start a treatment cycle? Could the patient's body be a safe laboratory for screening minuscule doses, nanodoses of different medicines before you begin a treatment cycle? And where we got our motivation from is actually from allergy tests. In allergy tests, patients are injected minuscule doses of different allergens underneath the skin, and then you look for the response inside the patient's body. And of course, the doses here are low enough so they don't endanger the patient if he or she have an allergic response. Could we mirror this concept and actually conduct it inside the patient's tumor and metastasis using nanoparticles? And what we did is we took 100 nanometer liposomes with a formulation very similar to the Doxil formulation, and we loaded them with minuscule doses of different medications. So now each nanoparticle has a different medicine inside, and the dose is actually so low it cannot be detected using the chemical tools we have today. So we added a molecular barcode, namely DNA, double-stranded DNA, about 100 base pair long, which we can actually detect even at a single molecule configuration. So now we have a cocktail of these liposomes. Each one of them has a different medicine inside of them, and also a way to detect which medicine is in each and every one of these liposomes. It's that molecular DNA. We inject the particles intravenously, and it takes them about 24 hours to circulate in the blood and actually accumulate in the tumor and distribute between the different tumor cells. And at that point, we wait another 24 hours. At this point, some of the medicines work and some of the medications don't work inside of the tumor. We take a biopsy from the tumor, and we look at the cells coming out of that biopsy. Live cells coming out of the biopsy have barcodes of ineffective medications, and dead cells coming out of this biopsy actually have barcodes of effective medicines inside the patient's own tumor. More than that, we can look today at subcells or subtypes of cells taken out of the tumor, and we can predict how cells or different types of cells from the tumor will respond to each medication, and I'll show one example about that. So just a bit about the process. The particles are injected intravenously. They circulate in the blood, and we can see nice accumulation in the tumor region. These are the red regions are actually the liposomes that are stained red in this case. This is the tissue cut, or deeper if you go in, you can actually see a signal from each one of the cells of this is the nuclei and this is from the barcoded nanoparticle. Or if you take these cells out, you can see nicely the barcodes inside a triple-negative breast cancer cell. So the barcodes are inside, but now what does the panel look like? In the panel, we have live cells and dead cells that come out of the tumor. And the upper panel here is of dead cells that were taken out of a tumor. And you can notice that the abundance of the blue barcodes is greater in the panel of the dead cells, the upper panel, compared to the blue barcodes that are in the lower panel, which are the live cells. Meaning we have more gemcitabine, which is encoded here with the blue line. We have more gemcitabine in dead cells compared to gemcitabine in live cells. And they're on for each one and every cell inside the system. Specifically, if we break it down medicine by medicine, when we took out the cells from the tumor or the biopsy and we looked at it, we found for each 5 dead cells that had a cisplatin barcode inside of it, we found only 1 live cell. So cisplatin here is effective. 5 to 1 is a good ratio. However, when we went on and looked at gemcitabine, for each 6,500 dead cells, we found only 1 live cell. So in this case, gemcitabine will be more effective than cisplatin, which will be more effective than doxorubicin, which also makes sense in the case of triple-negative breast cancer, where sometimes it doesn't respond well to doxorubicin itself. Taking it one step further, we created a potency chart which actually ranks the activity of each one of the medicines inside the tumor. So gemcitabine comes first, then cisplatin, and then doxorubicin. And the reason is to make the information actually usable to physicians who will make clinical decisions later on based on such a technology. We wanted to check that our prediction, if it actually mirrors out also in the therapeutic outcome. And you can notice that on the right-hand side, we treated tumors against the prediction with doxorubicin. The tumors are larger compared to the tumors that were treated on the left-hand side according to the prediction. Yeah, this is, I think it's dead. But not only the size was actually different. When we looked into the tissue itself, we saw a great difference between tumors that were treated according to the prediction, or tumors that were treated against the prediction. On the right-hand upper corner, this is a Ki-67 marker, and you can notice that tumors that were treated against the prediction were actually very vibrant and actually in a metastatic state compared to tumors that were treated according to the prediction here with gemcitabine that are actually going into regression. So not only do you see the size actually is different, but also at the micro scale of the tumor itself, we saw a great difference between treating according or against the prediction. We wanted to take the technology one step further and look at different cells that compose the microenvironment of the tumor. And we know that a tumor is actually composed about 50% cancer cells and another 50% are stromal cells inside the tumor. Now, each one of these cells responds differently to the medication that is actually given to the tumor. Namely, the cancer cells may respond differently than the macrophages or the T cells to that specific treatment. And when we broke it up, the first thing we wanted to make sure is that our liposomes actually enter each one of these subcellular types inside the tumor. So we created a single-cell array of the biopsy, and we looked at different types of cells taken up. And the nice thing was to see that some of the cells, as we expected, macrophages took up more nanoparticles than cancer stem-like cells, and it pretty much mirrored what we thought would happen. So Some of the cells take up many nanoparticles, and others actually take very, very few. And those are the ones we actually wanna look at in each one of these subcellular compartments. And when we looked at activity, we saw that different medicines act differently on different types of cells. And this slide is a bit busy, but I would want to point out one application or one relevant outcome of this technology. For example, if you see that your T cells are responding strongly to the medication, meaning that you're depleting T cells, going back 2 lectures, we probably wouldn't want to give that medication to the patient together with immunotherapy. Meaning if we're depleting now T cell population, your ability to then go in with immune activating system will probably be contradictive one to another. And more than that, as a strategic tool, who do you want to go for? Do you want to go for the main cancer cells, or do you want to go to the stem-like cells inside the tissue? And I think those are some of the questions that physicians will be taking throughout the next years, more strategic decisions of what we want to target inside the tumor tissue. And more than that, does the primary tumor respond similarly to metastasis? And we looked at, we know that metastasis is one of the more challenging states of cancer when coming to treat. And first, we wanted to make sure that our barcoded nanoparticles actually reach the different metastasis. We created a spontaneous metastasis model. These are mice. This is a breast cancer orthotopic tumor. You can notice as time goes on, the metastasis actually starts spreading to the lungs and other regions in the body, the brain, and the bone, etc. So we went to each one of these sites and we checked how each one of them actually responded to medication, to the different barcoded nanoparticles. I won't go in, but we were first validated that our nanoparticles can really reach these sites of metastasis. The pharmacokinetics is about 24 hours after the injection. You see a nice peak in the different metastatic sites, and you can target even very small metastasis, namely smaller than 2 nanometers. You find your nanoparticles inside these metastases. But going to the activity, we actually noticed that the primary tumor responds many times differently than the metastasis when you look at medication. Here we had a slightly different panel of drugs, and you can notice here while the primary tumor responded primarily to gemcitabine, the brain metastasis was responding very strongly to a molecule called salinomycin, which was discovered by Bob Weinberg as a molecule that can treat different types of cancer stem cells. But what made me actually worry greatly about treating metastasis was this graph on the right-hand side. And it looks at the activity in the primary tumor of a single medicine, and we gave that the number 1. And in order to deplete a single cell, so we needed 1 quanta of medicine to kill a cancer cell in the primary tumor. However, to kill that same cancer cell when it's in a metastatic site, we needed up to 13 times more medicine when we went to treat a bone metastasis. So sometimes going for treating the metastasis is going to be a much more challenging objective. And of course, the physicians see that day in, day out. And maybe what it requires is actually more combinations of different medicines. I'll just end with this. Does this mirror also the clinical the clinical reality? We ran some PDX trials. They're actually ongoing right now. And the first thing we wanted to make sure is that we can actually target the PDX tumors after an intravenous injection. This is from 2 different patients. And you can notice that the tumors light up beautifully. This is with ICG-loaded liposomes 24 hours after injection. So very similar to our orthotopic breast cancer models. More than that, we were able to find our barcodes inside the cells within the PDX tumor. It's a tumor that looks very different than tumors we usually work with when we work with orthotopic usual tumors. And more than that, and this is a comparison between 2 patients. We don't have the full story here yet, but you can notice that while one patient we predict should respond to cisplatin, the second patient should respond better to doxorubicin. Also, the magnitude of response seems different in this patient compared to this patient here, and we're actually following up now and treating the mice with the different medicines in order to make sure we can actually predict what will actually happen inside such a tumor. I'll just end with a short animation that I think explains the technology far better than I can. These are the barcoded nanoparticles being injected intravenously. They circulate in the blood. They enter the tumor through the leaky blood vessels. They distribute between the different cells inside the tumor and metastasis, and insert their medication, of course, together with a barcode. And each type of cell will respond differently, actually, to different types of medication. So I'll wrap up by first thanking you for taking the time. It's a bit overtime to listen to me today. We use barcoding technology in order to detect single-cell drug activity inside the tumor. What we notice is that subsets of cancer cells in the microenvironment actually respond differently to medication. And when you come strategically to treat, you probably want to know how each subcell will respond, and then go for your main target. Metastasis is very different. The way the metastasis actually respond is different than the way the primary tumor responds to medication. I'll end by thanking really amazing students that I enjoy interacting every day, and the funding bodies that help support our research. Thank you so much for taking the time.